Hi guys, welcome back to the second video of gene cloning from the subject of genetic engineering. So we'll continue from the previous video where we left from the diagrammatic representation of a vector. So as we know from the last video that a vector has three parts. So it has the origin of replication, secondly it has a multiple cloning site, and thirdly it has a selectable mark. Alright, so these three parts are very much essential and mandatory for a vector to have to act as a cloning vector. Alright, so moving on from there on, let's move to the next slide. As you can see, it specifies a palindromic sequences in DNA. So what is a palindromic sequences? Palindromic sequences are nothing but complementary sequences which restriction endonuclease enzymes recognizes in a particular stretch of sequences so that it can cut and thus a foreign DNA can be ligated. Alright, so coming to the second restriction enzyme, which is the HIN3. So HIN3 also forms formation of sticky ends and recognizes palindromic sequences and thus cuts this way, same as EcoR1 as you can see, but just the sequences are different and everything remains the same. And talking about another restriction endonuclease enzyme, which is the SMA1. Alright, so SMA1 does not produce sticky ends there, whereas it produces blunt ends. You can see everything is same in this, also everything is same in here. Alright, so everything stands complementary, right? So it does not lead to sticky ends just like in HIN3 and EcoR1, whereas it is blunt in both the ends, whereas it's blunt, and thus its recognition pattern is this way and not the other way around. Alright, so hope you can get these ways. So let me just highlight these things. So as you can see, this is the recognition pattern in this, and whereas this is the recognition pattern for this. Alright, so moving on from there on, you can see a sticky ends formation of here. So this is the pattern, general pattern for EcoR1, whereas HIN3 recognizes this way, the same as EcoR1, whereas SMA does not form this way. As you can see, I'm trying to draw this. This is the pattern for this formation of sticky ends, but it does not. It leads to formation of this way sticky ends. So this is the pattern, depends on the different sort of patterns. Alright, so in this DNA ligase, alright, so ligation joining of DNA molecules together. Alright, so as you can see in this picture very clearly, alright, so this is the vector. Alright, this is the vector. Alright, so this part has been removed, alright, with the help of restriction endonucleases, which can be any EcoR1, HIN3, SMA1, and any other. There are lots of restriction endonuclease enzymes. Alright, so this part has been removed. Alright, and this part has been replaced by the gene which needs to be cloned or which needs to be required in large quantities, or which is the foreign DNA. In simple words, so this part is ligated into this, which results in this. Alright, so this part is ligated into this to form this. So this insertion or ligation of this is known as ligation. Simple words. This is simple from the words only. Simple. Also, here we can see a ligating blunt ends and ligating sticky ends. Alright, so as I told you, blunt ends are not that easy, but obviously these also get joined after all. But in fact, sticky ends are easier to get joins. So as you can see, the joining of blunt ends here, and whereas the ligation of sticky ends here. Also, at some times, ligation of sticky end forms discontinuities, as you can see. But though we prefer getting sticky ends instead of blunt ends, because it is easy and it is most prevalent in most of the bacterium and other vectors. So though it forms discontinuities at places. Alright, so also with the help of DNA ligase, there's another enzyme known as DNA ligase. Also, I just forgot to tell you that ligation is the thing which is done by DNA ligase and no other enzyme. Alright, so this is one of the important things that we need to know. So moving on, so this is the ligation mixture product. So the product of ligation, as you know, so you can see the ligation products. As you can see, the things get ligated with the help of DNA ligase and no other enzymes. So DNA ligase is a very important enzyme that plays place in ligation. So this is all about it. So you can see it very easily, the cells containing wrong. So all of these is very much self-explanatory. So all circular molecules will be cloned, whereas the desired molecules will be cloned and not. So let's keep this video till this and stay tuned for more such videos. Thank you. So this is how it looks like. So it has a AB resistant or a resistant selectable marker, which can be ampicillin tetracycline or both. It has MCS or multiple cloning sites, and at the bottom you can see origin of replication through which it divides. So I want to keep this video short and simple. So I'll be explaining the further parts of this, how it divides and how the role of restriction endonuclease enzyme takes place in the upcoming slides. So stay tuned for more, and thank you for watching this video. So there are basic 5 steps that are involved in gene cloning. So the first step which comes in gene cloning which is the construction of recombinant DNA molecule. So it happens through a fragment of DNA which contains the gene to be cloned is inserted into a circular DNA molecule, which is also called as a vector. Don't worry if you don't know these terms, I'll be explaining in detail through a diagrammatic representation in the upcoming slides. So in this, basically our DNA is inserted into a gene through a circular DNA molecule which is called as a vector to produce a recombinant DNA plasmid molecule. So coming to the second point, which is the introduction of recombinant DNA to a host cell. So as we got the recombinant DNA from the first step, now we have to introduce this or insert this DNA into a host cell, which is a normal bacteria or any sort of bacterium that occurs in our environment, with the help of 2 processes. As you can see, there are 2 processes that are highlighted, which is the electroporation and CaCl2 or CaCl2 or heat shock methods. I'll be explaining all of the processes in detail in the upcoming slides. So coming to the third part, which is the multiplication of recombinant DNA molecule. So now as we have inserted the recombinant DNA to the host cell, now it will start to multiply within itself so that it can form as many number of recombinant DNA molecules within the host cell, which is the bacterium. So as it says, within the host cell, the bacterium multiplies, producing numerous identical copies not only of itself but also of the gene that it carries. Coming to the fourth part, which is the cell division. So after a large number of cell divisions, a colony or a clone of identical host cell is produced. So as you know, in the third step, there are many number of recombinant molecules that are formed through division. Now in the fourth step, cell division says that the bacterium divides into 2, and each bacterium has its own recombinant molecules, many recombinant molecules in itself. So it can form more number of recombinant DNA molecules in these bacterium. So now these have become now a single bacterium has become 2 bacterium through cell division, and now they can produce more number of recombinant DNA molecules. So this fourth point is relevant to the cell division point, which says the gene carried by the recombinant is now said to be cloned. In the red highlighted point, you can see that it is now said to be cloned. So coming to the 5 points, so how do we know that the particular DNA molecule is a recombinant one? So the fifth point, which says that the selection of the recombinant DNA molecule, which happens through selection of presence of antibiotics. So in this, antibiotics can be ampicillin or tetracycline, which I'll be explaining all of its importance in detail, or it can be other molecules in the bacterium. So it basically depends on the type of selectable marker present in the plasmid vector. So it depends on the type of antibiotic or the gene which is present on the plasmid vector. I'll be explaining what the term plasmid is also. So moving to the next slide. So yeah, so in this we will insert the bacterium. As the second point states, as the recombinant DNA molecule gets inside the bacterium, as you can see here. So the third step is the Before cell division, the recombinant DNA molecule that gets inside the bacterium gets divided or divides, and thus we get more number of recombinant DNA molecules. All right. So from that we got number of DNA molecules in a single bacterium. So now the fourth step comes, which is the cell division. So now, as we know, the number of DNA molecules have been formed, have divided. Now the entire cell divides. The entire bacterium divides into two. All right. So entire cell divides into two to form two bacteriums. All right. So it gets divided into two to form two bacterium, and these two bacterium individually, in these two bacteriums, the number of recombinant DNA molecules will divide in itself to form more DNA recombinants, so that the number increases, and thus we can get more number of antibodies through it. So in the fifth point comes the selection. How do we know that which of these recombinant DNA molecules are recombinants, or all of these which have reproduced or divided or formed more number of DNA recombinants belong to that particular bacterium? So there are a number of methods which we know as selectable marker, as I've said, selectable antibiotics such as tetracycline and ampicillin, which I'll be explaining in the further coming slides. So moving on from there on. So let's talk about a plasmid. What is a plasmid or a cloning vector? So what is a cloning vector? As I've previously termed that what is vector and all everything. So now you know what is a vector or a plasmid. So what is a cloning vector or a cloning plasmid? So basically that vector is known as cloning plasmid or a vector when it has the capability to attach or get inserted with the help of a fragmented DNA. In simple words, when a vector gets attached or gets ligated with the help of a foreign DNA or a fragmented DNA in the circular DNA, so that it can be inserted into a bacterium, and thus it can form divisions and as many as colonies it can form in it. So basically a cloning vector is a plasmid, which is a circular molecules of DNA that lead an independent existence in the bacterial cells. Alright, so the second point is plasmid almost always carries one or more genes, and often these genes are responsible for useful characteristics displayed by host bacteria. And the third point is the plasmid DNA can be used as a cloning vector. Definitely a plasmid DNA can be used as a cloning vector when it has the ability to get attached or get ligated by a fragmented or a foreign DNA, and thus it can be inserted into a bacterium, and thereon it can divide and form more number of DNA recombinants. Alright, so let's come to the components of what a DNA cloning vector must possess so that it can be called as a cloning vector. So the first point, which is the selectable marker, as you can see. So some of the examples of selectable markers are antibiotic resistant gene, which can be chloroamphenol, ficol, ampicillin, streptomycin, erythromycin, kanamycin, tetracycline, etc. Many sort of resistant gene. The second point, which is the multiple cloning site. So this is the cloning site for gene of interest, and it has the multiple cloning site and recognition site of many different restriction endonuclease enzymes. So I'll be explaining what are endonuclease enzymes and what all of these terms mean, and what are a restriction endonuclease enzymes. So basically this is one of the most important these enzymes which are involved in gene cloning, involved for the ligation and cutting and everything, which I'll be explaining the further coming slides part. So third point is the origin of replication. So most plasmids possess at least one DNA sequence that can act as the origin of replication, so they are able to multiply within the cell independently. So we have seen the previous slides, so the cell divides and number of recombinant molecules are formed in large numbers. So how does it happen? It happens through origin of replication. So origin of replication is involved for division. All right, so this is the main part which involves in the division part. All right, so I'll show you a diagrammatic picture of how a DNA cloning vector or a plasmid looks like.